welcome to another episode of Gray Matter. If you're like most people, you sometimes have trouble planning your day. Or if you're very organized, perhaps you can plan a week ahead. Or if you're really efficient and you've been reading a lot of books like 80-20 and the 7 Habits of Highly Effective People, maybe you could plan a month or even a year ahead. But could you imagine planning 500 years ahead? Could you imagine contemplating the secrets of the genome? Well, today we have a professor of genetics from the Ivy League School of Cornell University in the United States who's done all of that. He spends all of his time pondering these deep questions, and he's actually he's actually envisioned a future 500 years, a very exciting future, 500 years hence. His name is Dr. Chris Mason, professor of genetics from Cornell University. So we're going to get to him in a moment and find out more about him and sort of mine some of his best ideas about science. But before we go there, as we always do, we're going to frame our discussion with a few quotations. The first one is from someone who we often quote on the show, and this is Albert Einstein, popularly considered to be a great scientist. Chris may disagree. I don't know. I know some scientists are not fans of Einstein, but I chose this quotation because I think it applies here. He once was quoted as saying, "Imagination is everything. It is the preview of life's coming attractions." And of course, Einstein was very famous for using sort of the palace of his mind to envision things that do not necessarily exist or are not apparent in the natural world. And it seems to me that's something that Dr. Mason is involved in as well. Next quotation is from a science fiction author, one of my favorites. His name is Ray Bradbury. And he once was quoted as saying, "I have never listened to anyone who criticized my taste in space travel, sideshows, and gorillas. When this occurs, I pack up my dinosaurs and leave the room." And finally, someone who is something of a scientist, certainly somebody who's known for space travel, and that's Mr. Elon Musk, the richest man in the world. And he was quoted recently as saying this. He says, "If you go back a few hundred years, what we take for granted today would seem like magic. Being able to talk to people over long distances, to transmit images, flying, accessing vast amounts of data like an oracle. These are all things that would have been considered magic a few hundred years ago." All right, so who do we have today? We have Professor Christopher E. Mason. He's a professor director, WorldQuant Initiative for Quantitative Prediction, Physiology, and Biophysics at Cornell University. His expertise is in systems biology, functional genomics. We're going to find out what that means. And his current research interest, according to his bio, is new biochemical and computational methods in functional genomics, novel techniques in next-generation sequencing, algorithms for tumor evolution, genome evolution, DNA and RNA modifications, algorithms for genome engineering, international standards for clinical quality genome measurements and editing, integrated molecular portraits of genomes, and metagenomes for NASA astronauts. This is going to be fascinating, to establish the molecular foundations and genetic defenses for enabling long-term human space travel. Mm-hmm. He's winner of many awards, including the National Institutes of Health Transformation R01 Award, National Aeronautics and Space Administration Group Achievement Award, the Pershing Square Cancer Research Alliance Young Investigator Award, Herschel Weill Collier Career Scientist Award, Valley Foundation WorldQuant Foundation Scholar Award, Center for Disease Control Prevention Honor Award for standardization of clinical testing. And he's also written recently a very fascinating book that we're going to talk about. He's going to talk about and tell us about, and that at the end, in our reading list portion, we're going to recommend to you. Welcome to the show, Professor Mason. Thanks for being with us today on Gray Matter.
Yeah, well, a pleasure to be here. Thanks so much for having me and for the very nice introduction. It's a pleasure.
Yes, so obviously people who follow the show, and you may be one of them, I don't know, you're in the United States, so maybe you don't, we're based in Canada, but the people who follow the show know that we're great fans of science. We've had some very renowned scientists on the program, including, for example, Dr. Jay Bhattacharya of Stanford, who's an epidemiologist and has been very much in the news lately. And so we love science, and we love to have people like you on the program so we can learn more about some of the work that you're doing. In that regard, perhaps we could start with this idea of the molecular detective work for undiagnosed diseases program. This looks very exciting, where you're involved with Dr. William Gall and researchers at the NIHS's UDP to look for mutations and molecular changes that may explain the mysterious phenotypes of no known etiology. Would you like to talk about that? And perhaps maybe you could tell us you could give us sort of takedown out of the ether, a basic understanding of what a genome is.
Sure. Yeah, I'll start with the fundamentals first, and then dive into some of the clinical work and work we do and kind of detectives of the genome. But first, what is the genome? And that is basically, the recipe to make all of the cells and tissues in your body. And this is, when we all started out, we all began as one cell, so just as an embryo. And what's amazing about actually embryos and even genetics, the power of genetics, is that all of the instructions to make all of the cells in your body and to respond to your environment and to live are all in that very first cell. And so that instruction code, it's kind of like the kind of the handbook or the really the wiring diagram for your body is in the DNA. It's stored in the nucleus of that very first cell, sort of in the inside, and that is basically 3 billion letters of a genetic code that, if you imagine, a good analogy is very much like a book. It's this, it's an instruction set to make your whole body and to live. And that tells how one cell becomes two, and two becomes four, and four becomes, really tens of trillions of cells that you are today. And so the genome, though, it's 3 billion letters long, and then it's, essentially some parts of it have genes which help make things like your hair, help make your enzymes, help make you so you can digest food, or essentially so that you can grow taller as you're younger. And so all those factors that you consider normal living are in the DNA. But the DNA is just the book as it's kind of printed. For example, if you have a book in a library, that's essentially if it was just all the letters condensed, you wouldn't know how to read it if there was no spaces or punctuation or commas or paragraphs. So when we first had the understanding of the genome, it was a lot like that. We just had letters. We knew we could see the code, but we didn't understand really the why the diction was a certain way, or why certain words came before or after another. Or even there was no punctuation basically when we first began to understand genetics. But now the genome is the collection of all those letters, and also the study of the genome is where is the punctuation, where's the most important word in a sentence that makes that cell turn on when you get outside and have sun, and you have to repair DNA, for example, which we see that in space flight too. So the genome is your personal recipe for creating your cells, and then how it responds is a very dynamic interchange between your genome and then how it's regulated and turning genes on, turning genes off, controlling essentially your genetic activity at a cell by cell level. So, and that's what we do in my lab, is we study the genetics and think about ways to sometimes tweak it, modify it, even improve it.
Is trying to organize and understand this genome, but also try to maybe this is the wrong word, to manipulate it and sort of, to use the vernacular, sort of hack into it. Is that part of what you're trying to do, and not for any sort of maniacal Frankensteinian mission, but in order to do things like to discover solutions for cures for disease and things like that?
Yeah,
Is that sort of what you're involved in?
Exactly. So sometimes the manipulation of cells is exactly what you have to do to cure them of a disease. When one of these bits of a genetic code or gene is wrong and mutated, let's say, and then creates a malformed protein, or creates a inflammation, or creates it so maybe your lungs don't form. All these are instances where the mutations in the genetic code or the damage to it can lead to cancer, can lead to disease. And so what's extraordinary is now we know enough of the genetic code, and we have ways to modify, even change it. Things that people have probably heard about called CRISPR is one of these methods where you can actually tweak the genetic code. Much like if you're typing a letter and you want to delete a word, you retype it. You can just do it very simply. We can now begin to do that inside human cells.
Wow, that's fascinating. So how is that practically done, though? How
So
do you manipulate something that's microscopic?
Yeah, you have to. Well, the first thing you need to do is know what the genome is and sort of where the targets are, and then once you have that sequence, none of this would be possible without actually having early work in the early 2000s that mapped and sequenced the very first genome, the first 3 billion letter code, and then now we've compared that code to other people who have cancer or who are healthy. You can now sequence the genome and compare it. So once you have that first map of the first human, you can then compare that to yourself and then see where there's any slight differences. And then you basically design a small piece of almost like Velcro that has what's called a complementary sequence, meaning it perfectly matches some spot you want to modify. And you can design these guide RNAs, and there's an enzyme that comes on top of it, which is called Cas, or Cas9 is the one we often use, but there are others. And it comes in and basically can create a break of the DNA, and then that can get replaced with something else you'd like to put in there, either for a single letter change, what's called a SNP, or you could actually change whole chunks of DNA and rearrange them. So this is through enzymes we discovered in bacteria, actually, that they use to have a memory when a virus infects them. They'll break it and keep a copy of it and integrate it. And these systems, called CRISPR systems, are what we can now use therapeutically inside human cells.
Wow. It sounds as though since the discovery of this genome, like you said, in the early 2000s, that the science that you're involved in has been accelerating very, very quickly. Is that fair to say?
Absolutely, it's been dramatic. Actually, I like to tell everyone in lab that every day they wake up is the best day to be a scientist and a geneticist, because every day has the most possible data that's ever existed and is creating more data continuously. So it means quantifiably and empirically, every day is the best day to do my job. It doesn't mean I'll have the best day necessarily, but it means at least the capacity is the greatest for discovery, innovation, and sort of knowledge application. Every day I walk in the lab.
Wow. I want to move on and talk about something else here that is very exciting. Now, we've heard about the Stalinist 5-year plans, but you've got a 500-year plan, and there's something called the Mason Laboratory. It sounds like it's named after you, and it's working on a 10-phase, 500-year plan for the survival of the human species on Earth, in space, and on other planets. And it says, to that end, we develop and deploy new biochemical computational methods in functional genomics to elucidate the genetic basis of human disease and human physiology. So, could you explain about that? Why the 500-year plan, and what is this all about? And I see here it's got 10 different phases, and then there's an end phase as well.
Yeah.
So, first, could we start with why the 500-year plan? How you sort of came up with this? It sounds like sort of a Einsteinian imagination problem here. Could you explain how you came up with this?
Yeah, happy to. So I certainly am not on the level of Einstein. I just was thinking ahead a little bit, and I've always been thinking a lot in terms of what will happen to people if you look out long enough, and thinking about what happens when the sun runs out of fuel, what happens to the Earth. I've thought about this since I was a kid, but that was just knowing that at some point the sun will actually become so bright it'll boil the oceans, it'll engulf the entire Earth, right? So in my head, I've always thought, well, we have about 5 billion years before that happens, which is a long time, but it's not an infinite time. So it's I've been thinking about that ever since I learned that fact, since I was a kid. And as I got more technical acumen and more tools built under my belt that we could use in genetics, it then really dawned on me that we don't have to just accept the fact that the sun will run out of fuel and we'll all get charred. We could begin to go towards other planets. We could actually eventually even leave the solar system, but not even that we could, but actually that we have to eventually. Otherwise, everything we've ever done and valued and built will all be gone. So I think that got me thinking. So I've known that since I was a kid, but I thought now it made me really think also that there's a purpose for our species. Like, people just have a hard enough time finding a purpose just for their own life or what they want to do for their career, but there's never been something that's been defined as really what's the goal for all of us, for our species. But thinking about on a cosmological scale, this becomes to me quite evident, is that we're the only ones with awareness of extinction. Like, as far as we know, we're the only life in the universe on this planet. Is the only place where life has been found. Maybe it's the only life that there is, and life is extraordinarily rare in the universe, and it's precious to begin with, because it's one of the few things that counteracts entropy and actually organizes the universe and can create consciousness and have people have dreams and make poetry and music, all these things we take for granted, I think is extraordinarily valuable and unique. And so, but we're the only ones that know that it's in danger. So I started thinking about this plan because I wanted some way to, really embolden people to think far ahead, which I think is a very human trait. If you think far ahead, even if you think one day ahead, that's something that humans only humans do on a real planning purpose, like intergenerationally or even days ahead. Many animals build nests for offspring, or they do a lot of complex behavior, but none of them build at an intergenerational scale. So I think that, again, all this gets around the fact that humans are extremely unique and I think worth preserving. And so I thought, well, we should use the technology that's come to bear that we've now used to plan multiple stages to understand the genome as the first phase, which actually just ended last year. So begin a basic understanding of the book of life, then to begin to modify it, tweak it, and eventually make it so humans could, in the later phases, actually get to other planets and eventually get to other stars. And so the 10-phase plan is the various steps we take to do human genome modifications, engineering, and even intergenerational clinical trials, basically. And so this is what I laid out. I go through each part of it in specific times, but it is, I think, our duty as a species that we have the unique capacity to be aware of our frailty of life and of all life, but we can use that to preserve life and serve as the shepherds and guardians of it.
When I looked at this, Professor, it was very interesting to me because, of course, as you talked about, you are a scientist dealing with things that are microscopic, but then you've extrapolated out from that this literally macrocosmic view of human existence and human survival and human flourishing by sort of building out from the genome and where this could take us. So you talked about the first phase. The second phase I see is supposed to last until about 2040. You want to talk about that a little bit? What the next step is?
Yeah. So in that phase, we actually begin to do some of the real, heavy modifications of human genomes, and also try and build what are called, genetic insulators, or ways where you can package pieces of DNA or genes that have functions and have them only be turned on at very specific times or in certain environments. And it's what I said, later stages of what's called synthetic biology. So this is where we are today. We're basically in phase 2, and some of the trials I was alluding to is that you can actually take a disease gene and actually repair it, in a patient. You can actually have homing beacons go in and modify cells and basically repair a damaged gene, or in the case of sickle cell disease, you can actually turn back on a gene that was turned off when you were younger. And so there's one of these clinical trials that was published, really amazing results last year, is when we were all younger as a fetus, we have a fetal hemoglobin. So hemoglobin is what carries oxygen in your blood. And when you have adult hemoglobin, if you have defects, that can often cause beta thalassemia or sickle cell disease. So the idea there is, well, if you had a functional hemoglobin that worked fine when you were younger, like as a fetus, why not just turn the gene back on and see if that could be used therapeutically? And that's exactly what's happened, is that turning that gene back on is what actually has enabled this cure of these diseases. So it's extraordinary to think about these beginning stages of tweaking, basically genomes in humans, and then begin long-term trials to see how they do.
What are the implications of this in terms of human longevity? Is there an impact?
Definitely. So what's interesting about human longevity is the average lifespan has continually marched forward, now getting up into the higher 70s and lower 80s in some countries, but the maximum lifespan has not at all budged in really centuries, right? So this is still about 126 is the maximum truly likely real recorded average maximum lifespan. And we keep getting a lot of people older, but no one has gone past that. And that's with all the advances in medicine in the past 50 years. The powerful thing about these genome modification tools is they may finally let us actually remove some of the limitations towards living a little bit longer, and certainly even cure and fix disease and discover disease before it gets too late. So there's a lot of advantages to the tools that we use at the hospital and that are coming online.
Do you find that you face because obviously this is cutting edge stuff, and some people tend to be fearful or distrustful of things that are new. Do you find that you've run into some controversy or criticism over any of the research that you're doing? Sort of messing around with this genetic code, that sort of the unlocking the secrets of human existence?
There's concerns. I've heard people say, are you playing God, or how do you know you're going to do things right? What if you do something wrong? Will this lead to a zombie apocalypse? All sorts of questions like that. And I think, the track and the means by which we deploy these technologies are the same things we do in clinical trials. We do it slowly, we do it carefully, and do it with only a few people to start, then, larger groups of people, and eventually you expand it. So we do it as responsibly as we've done any rollout of medicine, which is imperfect historically, but it is the best way we know to actually get medicine out and to cure people. And so I think when people say, "Oh, this is curing a disease," there's less criticism, less concern. But if it's just for an enhancement or a modification, then there's questions of, well, do you really need to do that? And that does have to be, I think, incorporated into a broader view of society. How much does something cost? What's the impact? Also, what's the intergenerational impact, which I talk a lot about in my book. And how do we measure that and account for that? So I think, as soon as someone gets sick, they generally are less worried about the implications of it, if it'll get them better, right? But beyond that, there are some questions that you have to keep an eye on.
Right. I don't want to get ensnared into the whole controversy around the COVID-19 vaccines, but is it correct to say that in terms of discovering new cures for diseases and developing new vaccines, that this mRNA and this new technology, if we can call it that you're working on, that is going to be really the new era, the new feature of cures and vaccines that are going to be commonly developed by scientists?
They already deployed somewhat in hospitals and patients today, and I think, yes, we will see more of this. And even the mRNA vaccines are really, in the history of medicine, among the most successful defenses we have against a disease. You have something that's 95% or 90% successful in preventing, death or disease is really extraordinary. So I think, but I think we'll start to see more of these kind of methods to not just make a primer for the immune system to learn about a pathogen, but actually to teach it more directly, or to modify the immune system after an infection. So, yeah, absolutely.
Okay. I want to jump ahead a little bit to some of the kind of exciting features of your 500-year plan. So let's jump forward to phase 6. Now, this is actually not that long. This is not that far away. 2060 to 2100, hopefully is in my lifetime, is probably in yours. Begin settlement of other planets and genesis of synthetic genomes. What's that going to involve? That
That's
sounds
where
pretty exciting.
Yeah, so what this means is we can start to go to Mars, and we're supposed to have boots on the red planet by 2035, maybe 2037, depending on who gets there first. China and NASA are in a race, and SpaceX, well, Elon says that we'll be there, in 2 years. That's going to be a little bit fast, but there is a race ongoing, a new space race to get to Mars, and so, but we want to go not just to visit, but to actually maintain a permanent settlement there. Same with the moon. NASA's the Artemis missions are going to be flying to the moon, and we're going to stay there. So it really is something people dreamed about, I think, several decades ago, but it was far too expensive. There was far fewer options of ways to get to space, whereas now there's actually many options on ways to get things into space, and also ways to get to other planets, or at least a handful, I should say, at this point. But it's really an entirely new era, and so what's exciting is we will, by 2060 and by the end of 2100, by reasonable projection, have people actually living on Mars, and actually maybe permanently there. Maybe even the first person might be born on Mars by that point, potentially. So we begin to see actually this come to fruition, a long held dream of humanity. And I think also the synthetic genomes, I mean, you can start to build genomes from scratch. We've spent a lot of time in the past 20, 30 years reading genomes, where we can see what's in the world around us, including in the world inside of us and our DNA. But once we have enough of these genetic maps and basically bits of genetic code, much like you would do if a DJ is mixing tunes, or if you're a painter and you're mixing colors, you can do the exact same thing, with genomes, and make synthetic genomes, make new ones from scratch. And so I think that's exactly what we'll see about that point. And we've even started to see some of this already.
So tell me about what panspermia is.
Right.
This idea that this is in phase 8, where you have a plan. Part of your 500-year plan is to begin seeding of Earth-like planets.
Mm-hmm.
How would that unfold?
Yeah, so panspermia. So this is in some sense also a big controversial question: is when, if ever, should you contaminate another world with life from this world? And this panspermia is the idea where you can actually send a sort of seed, hence the root of its sperm, as in like a basically germline tissue, to another world to basically seed it with life. And you could call it pan ovia. It doesn't have to be spermia. It doesn't have to be male, but you could have it be this future where we decide to say, okay, we found the best planet that looks like it's habitable potentially for human life. We want to begin to have things grow there before we get there, and essentially send a package of organisms that would adapt and survive and begin to grow on the planets well before we get there. So the danger with this is there's something called planetary protection, where if we're worried about us contaminating Mars or Mars contaminating Earth, say a super pathogen, what Michael Crichton called the Andromeda strain, and we don't want that to happen. But at the same time, again, I think all these questions become crystal clear through the lens of 5 billion years. And so if you think, well, I'm worried about contaminating Mars, but if you look out far enough in a timeline, if there's no life on Mars and no life anywhere else, and we are the only ones that can serve to safeguard life or to move it, at some point we will have to go through Mars to get out of the solar system. It's the closest place we could start to settle to get farther out. And so I think in a long-term view, planetary protection has to be balanced with really the needs of life itself. And so I think there is that's part of the goal there, is to begin to go towards the preservation of life itself. So I think panspermia is one way where you would purposefully put life onto a planet and start to plan to go there. But we'd have to do that, I think, slowly, and make sure that if there is life there, we try to work with it, not just totally decimate it.
Yeah, it's a tricky prospect, isn't it? Because of course nowadays the whole idea of colonization and imperialism is rather unpopular.
Yeah, of course,
and those of us who are fans of Gene Roddenberry and Star Trek, we remember the Prime Directive, which was, you're not supposed to go around the universe disturbing life that's already there. You're there to seek out, seek out new life and new civilizations. I guess the danger would be we could be going there to mess with life that isn't apparent to us, isn't evident to us, but could be existing. But I guess this is the really fascinating part about this is there's this tension between we want to discover, we want to reach out, we want to, there's an imperative for us, as you say, because our planet is dying. But at the same time, we have to be respectful of other life forms and the possibility that other life forms exist, and not to do anything that's going to harm or potentially destroy them, right?
Right. I mean, I think if anything, the Prime Directive actually, I think, makes perfect sense in a universe where life is abundant. But if we have several millennia of searching and there appears to be life nowhere, we would have to reconsider that Prime Directive and say, well, if there's, because I think it's not a bad idea to say, let's jump right in and immediately contaminate a planet as soon as we find life. Definitely not. But it would be that, if there's really no life anywhere, or if we go to a planet, for example, imagine this is thousands of years from now, and we stop by a planet and for all we can model and discern, it looks like the life is, say, bacterial life, but we think it will take several billion years for it to become complex life and never be able to get off that planet. But let's say the star is only a few 100 million years away from burning out. And we think, well, okay, the best way to actually preserve this life is to go get it or to push it along to become more complicated. So it's an inverted Prime Directive from Star Trek, but I would argue a more ethical directive, because the alternative is you just let life just die and you let it die to history and disappear. Whereas I think, that would be wrong if you assume life has value, but it assumes also that our modeling is correct, that we know what we're doing, which we can't do any of the things I've just described quite yet. But I think eventually we will build these models and actually have to face these questions.
Tell me about the importance of Mars. Of course, anybody who's knows about H.G. Wells is fascinated by Mars. The warlike planet and all of that. Why is it really important? What's the importance of Mars in terms of, let's say, a stepping off point for this type of space exploration?
So Mars is, in some sense, it's not the closest, it's not necessarily the best. It's just the one that's, about the best that there is, because it has at least some atmosphere. It has a similar day cycle in just about 24 hours. It's close, but it has a very thin atmosphere. It's very cold, but we can probably survive there. Whereas Venus is way too hot. Basically, it's hundreds of degrees Celsius. So you just like it can melt metal as soon as you get to the surface. It's way too hot. It's a runaway greenhouse effect. And we in Mars is it does have some caveats where it doesn't have a magnetic field, so it won't protect you against a lot of radiation from space. But on the other hand, you could go down, burrow into some of the the lava tubes, or these places where lava has come out from the planet, be somewhat protected from the elements and radiation, and build real habitats there. And so I think this is the closest and best bet that we have for getting to another planet and surviving there. Whereas anything else is farther away. We would take too long to get there, and we wouldn't be able to survive quite yet. So, but it's the first step on a ladder out of the solar system.
Right. And then in phase 9, we get into the shipment of humans to these new worlds. That's between 2301 and 2400. And then phase 10 is human settlement of a new solar system and used as a model for future systems. So how do you envision that unfolding? Or is that too far hence?
No, not at all. I think that's close in my mind. I feel it is. So the reason once we perfected some of the interplanetary work within this system, and even had people on Mars and maybe even Titan, we could then get to a place where we say we know we can survive in space and on other planets, but eventually we still have the same problem. The sun, well, we cannot stay in this system forever, or for that matter, any system. But we need to begin to go to a different one. And what you want to do is then say, okay, well, let's perfect the technology so that we could have what's called a generation ship, where multiple generations live and die on the same spacecraft on their way towards a new star. And, that raises a bunch of questions. Well, which star do we go to? How do we know it's got planets? What do you do for multiple generations that live and die on the same spacecraft? What happens to the people who are born into a spacecraft and they can't go anywhere else and they're stuck on a ship going just one direction? Would they mutiny? Would they get upset? Or would they feel honored that they're the chosen ones of humanity to go, populate a new solar system? So these are big questions. But overall, the technology by then would be perfected, and we would also know which planets to go to. Right? Basically, 25 years ago, there was only a handful of planets ever found outside of our solar system. Now we have over 4,000, and several hundred of them are in a zone called the habitable zone, which is likely having liquid water, where humans might be able to survive today for some of them. So, by the time we get to that year 2400, we will likely have hundreds of thousands of candidate planets to pick from. And we would then pick the best ones and send ships there to try and basically the generation ships would go. Multiple generations would live and die, likely, unless we magically perfect, faster than light travel or really warp speed or something like that. But in the book, I make no assumptions about large jumps in technology for travel. I say, let's assume that just stays where it's at. What can we do with the biology, and think about the ethics of it, and think about what we should do for our species and for all life. And so that's how we end up there. And then eventually we'd be hopefully around another star.
So what made the discovery of all these new planets possible?
A lot of this was something that was really looking at, taking telescopes that can look at the surface of the atmosphere of these planets. And you can actually see essentially, on some of them, you can actually get these emission spectra of what, elements are in the actual atmosphere. Essentially, when it passes in front of its sun, you can visualize that. But passing in front of the sun is really the big development that let us find all these planets and start to characterize them. Is that it's basically, once there's of course orbital changes when planets move around each other. If there's, of course, you can model, their orbital mechanics and see, are they close to another planet? Are they effectively being, is the gravity moving them? But you can also look at the luminescence, and so you can actually see that, if there's a bright star that's, blinking at us, and you can see the light normally, but as soon as a planet, especially a big planet, goes right in front of it, it dims the light just enough to actually see and say, aha, there's a planet there, and then you can map that out. And so this is what's called transiting. So you can see this as transits through the light. You can see that change. There are other methods that were used on some of the velocity calculations, like radial velocity is another method, but the transiting system and the transiting telescopes that watch this dip in the luminescence of the sun as a planet crosses in front of it have led to this discovery of most of the planets.
Is a lot of this research being done right at Cornell or at other American universities?
Lots of it. Some at Cornell, some actually at Jet Propulsion Laboratory is a large hub of the research there. Some at MIT. Sarah Seeger does a lot of the work there. So across the U.S., but also a lot of international partners are contributing to finding basically these new planets, which could be potentially new homes.
So this 500-year plan, even though it's your conception, you've got other leading scientists also working on this project with you. Is that correct?
Absolutely. Yeah. So many of us all working together. I'm coming more from the genetics and the biology side and looking at the planets to see which ones would be useful for life as we know it, but then thinking, in a creative way, how could it be matched to life as we could imagine it? Could we imagine life with new features or functions or forms, and basically create life so that it could survive there? And I think the answer is, we know the answer is yes, like to some degree we can and are doing this even in my lab today.
Okay, let's talk about the big one, the Armageddon. We've all seen we've seen the movies, the giant meteor, or however it happens, entropy. You have this as phase N, the end of the universe. You say the hardest question, determine if we should prevent the implosions or entropy death of the universe or allow self-destruction in the expectation that life will arise again. That's a very perplexing problem, isn't it? It's not just scientific, it's ethical.
Yeah.
How do we, can you explain that? How do we answer that question?
Absolutely. So this is really, because I talk about the end of this solar system and the need to eventually go. We have no choice really in the matter unless we figure out some way to survive on a giant sphere of fusion, which I think is unlikely. But we have to go to another solar system as a fact. But then it begs the question, well, if you go to that solar system, won't that sun run out of fuel? And you have to go to another one and keep hopping around between different solar systems. But it really just begs the question, well, where does it end? Ad infinitum? Do you just keep going on and on and on to a different star? And for a little while I think we would have to, but then if you take a projection far enough into the future, it's the big question of, well, at the end of the universe, what do we do? And that means the end of the universe will likely end up with sort of a heat death of the universe where it keeps expanding and expanding out and out and out, and eventually everything gets too far away, so you can't even see. Eventually, the sky, if the Earth was around that long, would be all black. Because all the light from other stars would be so far away, the stars would all be too far away for the light to actually reach us, because it'd be so just getting farther and farther away. So we imagine that period. It starts to become kind of a lonely and dark view of the universe, where everything is too far apart from each other. But so another thing that could happen, depending on the amount of dark matter and dark energy in the universe, is the universe could crunch back in on itself and recollapse and maybe create a new Big Bang. But in either circumstance, life would face a version of extinction, a permanent and universal extinction. And so the question there is, well, say we've mastered physics enough to warp space-time or restructure the universe itself at that point. Should we then actually, basically use our technology to prevent what would be the natural course of the universe, where it would keep expanding or implode, or just let it go and hope that life would arise again in the next universe? But we don't know if it would. And I think we would have to be consistent. We would have to preserve life, so we would actually try and prevent the natural end of the universe and make it so that we would actually survive it. But even then, we might not succeed, because if everything is really tearing itself apart and faster, we may not have a way to stop that. But I think if we can stop it, we should stop it.
Okay, fascinating. I watched the fairly lengthy interview that you did, another one that you did on another program, and there were some really great topics there, and I want to hit some of these. So we went from the end of the universe now to another one that has a lot of appeal, a lot of fascination for people, and that's aliens. Mm-hmm. So I want to start here. So you're probably aware, I'm sure you are, that the concept of UFOs and aliens is very, very topical. In fact, seem to be increasing reports of sightings of spacecraft and things like this, inexplicable things that are being seen by, for example, jet pilots and so on. First of all, so my first question is, do you think that there is other intelligent life in the universe, more advanced than us? Mm-hmm. And secondly, have you thought about what we would do or what we should do when we meet them?
Good. So, really good question. I think it's something we've all wondered about at some point. Either you saw it in a movie or you just look up at the stars at night. So I believe it's basically a belief question, because, of course, we have no hard evidence that there's intelligent life in the universe. There's been a potential spacecraft that came through a few years ago that went into our solar plane potentially has been talked about, or some of the UFO type objects that have been documented by the military. There's potentially some evidence that something's out there, but there's no really solid evidence where it's a clear communication from an intelligent life form. So, barring that, I'm going to say I don't think there is, to our knowledge, there's no intelligent life out in the universe that's there. But if I were to guess, is there any life out in the universe? I think probably, because microbial life has been found literally everywhere on the planet that we've looked. And if it can form, it seems like it will. And so I think, we've had a few billion years to make life on this planet, and it eventually was created. And so I think other planets around the universe are still cooling and being formed. But given enough time, I think at least microbial life probably would, and I think probably already has appeared somewhere else in the universe. We just haven't found it. We haven't obviously been to that many planets yet. So I think we'll probably find something like microbial life once we get out there.
Mhm. So what would we do when we meet them? What's our, getting back to this prime directive? What's our moral obligation? How do we do? We do try and kill them, or do we
No,
shake their hands? Or is it like Richard Dreyfuss? If you saw that movie.
yeah. So, and I think a lot of people are afraid. They're worried that we would either be the aggressor or the other aliens would be an aggressor. I think, and the evidence is that if you look on Earth, there's lots of struggle for resources between all species on this planet. It's not like it's a kumbaya universe here, but it is, a chance, if it's really an intelligent life form, to work with it, or to, collaborate. Again, we've seen in human history that sometimes we interact with other cultures, it has been, led to war or destruction, but then eventually could lead towards a harmony and collaboration. The U.S. and Japan were mortal enemies in 1941, but now are extensive collaborators. Not that long after, and so it could even start in a really antagonistic way, but then become very friendly and collaborative eventually. And if it's microbial life, I think we'd want to serve as these kind of guardians and stewards of life, much like we would for life on Earth. So I would hope that we would continue that goal and that mission, and maybe the life that we would encounter would already have that sort of sense of duty towards life. Let's hope, at least.
Okay. Well, I'm going to ask you a couple of these topics that I've written down here, and the kids have to leave the room right now because we need to ask you about sex in space. Not just sex in space, although that's very fascinating all on its own, but just the concept of procreation, because we know that being in space impacts the human body in ways that that require or cause adaptation. So talk about the human genome and the human body. How do you foresee that unfolding in terms of procreation, perpetuation of the species biologically, using your example of people traveling on a space caravan towards some unknown galaxy, unknown universe, unknown planet? How does that work? What are the challenges that are going to be faced under those circumstances?
Yeah, there has never been officially. There's never been sex in space, as far as we know. And there have been mice have been born in space, where pregnant mice were sent up to the space station and they gave birth. Some animals have had full reproductive cycles, like fruit flies or small worms called C. elegans. So we know it's possible to reproduce in space, but we have never really tested this for a mammalian system, from fertilization all the way out towards birth. And so it's not clear, and even just the act of having intercourse would be complicated in space because you're in microgravity. And so, as Scott Kelly said, you would probably need some straps and some restraints. That he said, not in a creepy way. And so we were recently talking about this at a session. I think you probably saw it, and it's fun to think about. But then you think, yeah, you would, because otherwise you'd float away, right? So it is,
Right.
it does make it difficult. And so that brings up, the idea of having artificial gravity with say, centripetal force, where one of the parts of a station would be spinning, so you would get some of the benefits of gravity in certain parts of your space station. So that would be one solution. The other one is to just, get lots of straps and figure it out in microgravity, or to get creative in some other ways. But this is a big unknown, because the radiation in space, the changes in gravity, lead to really a lot of unknown developmental challenges that we have not really teased out yet, and we are really hoping to do so, or to get more information about this when we start to get to Mars and get to other planets, and really kind of tease this out.
Mm-hmm. Well, maybe we could ask William Shatner. He seemed to have a very active libido when he was Captain Kirk.
Yes, right.
Okay, so what about culture living on Mars? How are we going to? You talked about preservation of culture, and that's part of what the 500-year plan is all about, trying to preserve what we've built on Earth from extinction, the best parts of it, hopefully the best parts. But how do we sustain that on another planet like Mars? How would that work?
So, I mean, what's amazing is that it would be sustained, but then also it would change. Like everything that happens with culture, you would get different dialects, you would get different traditions, different ways to eat the same food, but with a different, spice maybe. So I think life continues to evolve, and so does culture. And so I think we would eventually see a Martian culture, much like even in the United States. There's people in the north and people in the south have a southern accent, so you get different accents, different cultures, different foods, different music. I think it'd be this wonderful blossoming of creativity under a new environment where
Mhm.
culture has always adapted and always changes every time humans have gone anywhere. So I think we'd see that in spades and probably accelerated on Mars. So I think people would want to carry their traditions. So, for example, I talk about in the book, if you're Muslim and you want to pray to Mecca, you're supposed to pray towards Mecca. But if you're on Mars, it might be above you or below you. You may have to figure out, where it is. Or do you still pray to that Mecca, or do you make a new one on Mars that's like a parallel one? That's a question for the imams, I suppose. But this is raises these interesting questions of how do you have religion on a different planet? How do you have, do you would you still have the Sabbath, or do you just not have it at all because you wouldn't necessarily have 7 days on different planets, right? So
Right.
it would force people to change their religions, their food, their culture, their music. I think it would all be changed, but in a really interesting way.
What about the concept of a chlorohuman? What does that involve?
It was one of my favorite discussions we had in lab a few years ago, and I said, it came up in just a thought of I just wonder if it's one of these things where whenever you see, or at least when I look at life, I see life that has all these features on Earth, right? You see tentacles, you see the ability for organisms to fly, and that inspires people to want to fly and build planes. And of course, that's already been happening for a long time. But I get even, I'd say, genetically jealous. I get really interested in thinking, well, I would like to have that trait. I see an organism that can do something like a plant that can just, if it needs food, it just unfurls its leaves and collects sunlight, and that makes its own food. And so just thinking, well, what would it take to make a chlorohuman? Make it so you have green skin. So if you get hungry, you go outside and lay out into the sunshine and fill up on sunlight. And how much skin would you need if you only want to lay out for 1 hour? So it's like 2 tennis courts worth of skin. I did the math in the book, which is kind of fun to think about. If you only want to be outside for like an hour, so it's you could unfurl your chloral green skin and then absorb and make a big and then go back inside, go in the shade and say, I'm all full, or something like that. So that's just in terms of how much surface area you'd need. But biochemically, it would be complicated to get it to work inside of a human cell. That's why it's later in the 500-year plan, because I think it'll take some tools we don't yet have.
That's very exciting. So another one I want to ask you about is clones. This is something that is very, very intriguing to people. This idea of clones. What is the role that cloning would play in this?
So I think cloning is something that people often are confused about or even terrified. They think of Dolly the sheep being cloned, or clones of dogs, or pet cats or other pets. But clones are actually very common. When you have identical twins, they're basically clones, right? There's one embryo that's been split into two, and it's a clone. And clones have also been made from even your skin cells. You can take one cell, basically treat it with what are called Yamanaka factors, reprogram the cell, and then make it into an embryo and make all your cells all over again. So this idea of cloning is actually something that is in some ways very common, in the sense we have twins all over the place on this planet for many species. But it's also something that's very powerful, because it means that any one of your cells can truly be the guidebook for making all of your cells all over again. So this is for maybe replacing your kidneys or your heart or your lungs. A lot of work in the regenerative medicine field is to do this reprogramming and variations on ideas for cloning to make the cells that you need, and even repair yourself with your own cells and your own genetic code. So this again is something that will become more and more common, I think, as we leverage either the tools of cloning or even clones themselves potentially. So yeah, this is again something I don't want people to be afraid of, even though it seems peculiar. It's actually very common.
It does raise kind of a tricky ethical question, though, doesn't it? In terms of what is the clone precisely? Is it a human being? Is it human life? Of course, this is very topical in the news right now with the whole concept of abortion and what is a fetus? When does life begin? So, what precisely, what rights would a clone, for example, have inalienable human rights? Or would they be treated as a sort of a lower species that could be harvested in order to make other people? It's a tricky question, isn't it?
Well, for the clones, some parts can be tricky, and some I think are very straightforward. Like for would clones and should clones be afforded the same rights as other humans? It's an unequivocal yes, as long as they're a sentient organism with ability to direct its own life, and is they would just be like identical twins. Like, do you give only one twin rights and not the other one? Of course not. They both get rights, and they're individuals, and they can vote and have jobs and have lives. The same would be true of clones, and so and there, I think people's intuitions about this will change. It used to be that, for example, IVF or test tube babies, everyone was terrified that they would be mutated, abnormal, they'd have problems, they wouldn't be like real humans. But now it's decades and decades later, and they're all just fine. And so I think we'd see clones would eventually maybe at first be treated that way, but eventually be just viewed as normal. Actually, now
Mhm.
almost 2% of births in the US, or almost 3% are IVF, actually. So it's very common. And the what you do to them though could change. If you modify the embryo of a clone, it might be sort of an enhanced human, and that might be more controversial for people. They might be treated differently, but they might also look different, right? So I think that's many, many years in the future. We don't have to worry about that yet, but the technologies are emerging that could make that possible. And so I think I would want to make sure that they have and that they're afforded all the same rights as other humans, as they should be.
Another topic that is of interest to many people is the role of AI, artificial intelligence. So, can you explain, or is there a way that artificial intelligence can, in some way, be fused and be made part of the genome?
It can maybe eventually. There is something described by people in the field, like Ray Kurzweil, someone who talks about the singularity. It'll be this moment where we can upload our brain into the cloud and live forever. I don't know if that will happen necessarily by 2036, is what the estimate was. It might take longer. But even just having AI embed in our lives and help us already happens today with most of the work on algorithms that run computers or cars or many of the financial algorithms. This is, again, very common actually already, but to really make it a true cybernetic and AI human interface would require things, new interfaces between our brain and some of these technologies. But I think I would welcome it because it might help us survive in places. It might help us actually live longer or better lives. And so I think it's a ways off for that, but I would welcome it. And even AI in general, some people are afraid that machines are going to kill us all. What if they machines become sentient and want to work with us and hang out and have beers with us? We don't know what they're going to do. But if you I think people that are afraid of AI either haven't done programming or haven't thought about what programming could be, that they could be benevolent, they could be even better than us. And again, it could go south, they could be bad, but I think they don't necessarily have to be. It really depends on who did the coding.
Um, Professor, I wonder if you could provide some advice to young people, students, maybe high school students or college students who are taking this in. If they're listening to you and they're learning about your research and they want to find out more about it, maybe get involved in this type of work. What sort of advice would you give to them in terms of where they can find more information or what maybe courses of study they could pursue?
And actually, there's a lot. The best thing to do is to read as much as you can, as often as you can. Become a sponge for knowledge, and then also, especially if you're talking to a lab like my laboratory or really any laboratory, you read the papers from that laboratory and you know what they've been working on. So when you go to talk to people in the lab, you know at least what they've already done that's published, and you can have an intelligent conversation. And yeah, it might be tens or hundreds of papers, but it's something you could do if you really care. Read and read about the lab that you're looking at. But there's also lots of courses that are online that are free. There's Coursera courses that are free. MIT has a lot of courses online for free. A lot of my courses are online for free. And then also there's NASA. If you like some of the work with NASA, they have a program called Gene Lab, which is a place where a lot of the data that's from space and other animals that have flown in space is there. You can play with the data, download the data sets. The other big advice beyond reading is get working at coding and analysis, because at the end of the day, that is one of the most precious skills you can have, is to be adept at processing data, analyzing data, understanding spaceflight data and biological data, because once you understand it and how the biology changes, you can then begin to model it, change it, even engineer it. But you first have to understand it. So I would say play with some of those data sets that are online, take some of those courses, read as much as you can, and don't be afraid to reach out to PIs, such even myself. Just say, hey, Dr. Mason, nice to meet you. I'm curious if I can stop by the lab for a summer or say hi. And the other thing I'd say is actually the last bit of advice: be persistent, because sometimes I get 600 emails a day. I might miss it or get buried. So never feel bad if you care about something to follow up and say, hey, I'm still interested in this. Is there anything I can do? Or do something and then say, I already did this. What do you think of my analysis of your data? That's even better. So be persistent is the last bit.
Well, this has been a really fascinating discussion, Professor. We're so grateful for your time. I've certainly learned a lot. I think many people, I expect anybody listening in has learned a great deal. This is the part of the podcast where we go to something called the reading list.
Mm-hmm.
And I'm going to go first, and then I'm going to let you finish off. It's obvious you've read a lot of books, but we're going to start with one recommendation that I know you're very familiar with. It's this book called The Next 500 Years, Engineering Life to Reach New Worlds. This was just published this year. It's your book.
Mm-hmm.
And in it goes into much more detail than what we've been able to do today about this plan for the next 500 years and how your research really explains how this is all going to unfold. If you were going to describe your books just sort of in a synopsis way, how would you do that?
Yeah, I would say, for the book, it is a roadmap for all the basic knowledge of biology and how a body responds to spaceflight and living in space for a year, for example. And then also not just what we know about biology. So it's really a tutorial on all the cutting edge science that's in my lab and many other labs, but also, what can we do to tweak it, to modify it, to make like chlorohumans or to modify microbes or to really enable us to survive on other planets. So it's a treatise of hope, I'd say, of what I think we can and should do, as well as a lot of fun science and some bits of comedy in there as well, I'd say.
Fantastic. So I've added one more book. It's science fiction, and you mentioned earlier Michael Crichton, who's actually one of my favorite authors. I've read all of his novels. You mentioned the Andromeda Strain. Of course, he's best known for Jurassic Park. But the book that I've mentioned here that's science fiction is not one of Michael Crichton's books. It's because we were talking so much about Mars. It's The Martian Chronicles by the late Ray Bradbury, who was mentioned off the top. And so this book actually envisions what life would be like on Mars, and it talks about many of the challenges that would be faced if we came into contact with Martians and how that would be worked out. And like all of Ray Bradbury's books, it's beautifully written, and it goes into a lot of it is science fiction, but there's also a lot of science in it. I hope you'll agree.
Yeah.
But so I'd recommend that one just as a sort of to take the thought experiment through of what it would be like to actually go live on another planet and how the relationship between humans and Martian life would unfold. So do you have some suggestions for us besides your own book, Professor?
I would say, yeah, actually there's several books that are really great. Kim Stanley Robinson has written these beautiful books. One's called Red Mars, Green Mars, Blue Mars, about terraforming Mars, that are well done. He's written a lot of other great books as well. One by Neil Stephenson is called Seven Eves. Highly recommend. It's very also intriguing view of the future of humanity. The other thing I would actually encourage people to read is a book recently I just finished last week. Is called The Precipice by Toby Ord, which is a lot about understanding existential risk to humanity and how do we address it. And I think we encourage people to just really be excited about what is the future ahead, because I think it really is actually quite bright. I think some of those books highlight the risks, but also the promise.
Well, thank you so much for being with us today, Professor. It's been a real pleasure meeting you and learning more about your work. Wish you much continued success in the work that you're doing. And I'm looking forward to at least the next part of the next 500 years that I get to see. Congratulations on the work that you're doing, in uncovering the secrets of the human genome.
Great. Well, thank you so much, and pleasure to be here. Have a great weekend.
Thank you.